Hello, my name is Logan Ratliff, and I am here to discuss my final cellular biology project of cystic fibrosis. Cystic fibrosis, an overview of what cystic fibrosis is a genetic disorder that causes thick, sticky mucus to build up in the lungs, digestive system, and other organs, and can lead to serious health problems and eventually death if left untreated. The cause of cystic fibrosis is via the mutation of the CFTR gene, which encodes the CFTR protein that can vary in functionality. This functionality of the protein and different classes of this protein will be discussed in the next slide and its severity. So, what CFTR stands for is a mouthful. It represents cystic fibrosis transmembrane conductance regulator protein. It's a mouthful. Otherwise, we just call it CFTR protein. And this protein regulates the movement of chloride and sodium ions across the epithelial cell membranes. And how this overall contributes to the general function and within the body is overall it helps control water balance, regulates mucus production, and clears mucus efficiently and effectively within the lungs if it starts to build up um, within the lungs. So the impacts of the proteins it can produce, as I said, there's different classes and different severities that um, can be associated with how the protein is functioning, rather. So the cause, as mentioned earlier, is a mutation in the CFTR gene, which encodes the CFTR protein that can vary in functionality. So class one is simply no protein is made. A class two impact can be misfolding of the protein. A class 3 impact can be the gating defect, otherwise discussed as protein when the protein reaches the membrane, but um, the channel that it interacts with opens poorly or slowly or not correctly. And then a class 4 impact would be reduced communications through the channel. And then a class 5 impact would be the reduced amount of stability of the CFTR protein at the membrane. So the impacts that cystic fibrosis can have, narrowing it down to the metabolic pathway and how the metabolic pathway is kind of impacted. Um, so firstly, it can impair the trans-epithelial chloride and bicarbonate transport that alters homeostasis, pH, and secretions, which makes sense because it can alter um, the overall view. It can alter mucus production. Um, because of that, it can impact our cells by dehydration of the cells and viscous secretions due to lowered activity in the CFTR channel. pH is also altered by the impairment of the bicarbonate transport that affect enzyme activations. So if our enzymes work at a certain pH, a little fluctuation within the pH um, can impact their function, and bicarbonate has a huge impact on our overall pH, therefore the activity of enzymes. And then lastly, a last metabolic pathway that cystic fibrosis can have is that the CFTR mutation impairs CFTR folding, so the protein itself folding and trafficking, which can lead to fewer channels reaching the cell surface. So, kind of widening our scope of what cystic fibrosis can lead to within the body, um, or specific organ system complications. I kind of touched on how specific organ systems are impacted, but here we can get a little bit of a more broadened or narrowed view of what organ system is impacted. So here we have the lungs. The lungs are probably the most impacted by cystic fibrosis, and that's because thick, sticky mucus can build up within our lungs, specifically in the alveoli, and cause obstruction and can cause chronic bacterial infection. So think about it: if you have a very thin lining within our alveoli that allow gas exchange, oxygen to come in, and then CO2 to come out. If we have mucus blocking or in the way of that, or gas exchange can't really get through as efficiently. Therefore, one may feel out of breath, always coughing, trying to get that stuff, out of us. So that's an impact within the lungs. If left untreated, that can be a very serious complication with that could ultimately lead to death. Within the GI tract or the gastrointestinal tract, intestinal obstruction syndrome and biliary cirrhosis from buildup of bile can occur. Our sweat glands can be impacted, and kind of makes sense because reabsorption of chloride leading to elevated sweat chloride levels can be a impact of our organ systems. And oddly enough, this is one of the tests that we will talk about later on that they use to test cystic fibrosis. And then antimicrobial activity. So because the pH is altered, our immune cells work at a specific pH, and if the pH is altered within the environment that they work in, they can therefore not function properly and not kill bacterial infections that are taking place within the lungs or wherever in the body. So, specifically discussing the heritability of cystic fibrosis, it is a autosomal recessive trait, and this occurs. It is a pathogenic variant of the CFTR gene on chromosome 7, and how this affects two individuals that may be carriers, who are carriers and want to have a child. Each pregnancy is about a 25% chance of an unaffected child, 50% chance, though, that the child is a carrier, and then a 25% chance that the child that they may have is affected. Within the cell cycle, how this is related within the cell cycle, when the people are produced, or a newborn is produced, it's not a failure of the cell cycle checkpoint. The defective cystic fibrosis relates to the CFTR protein production, which misfolds and processes the protein. So this does not um, lead. This is not because of a mishap in the cell cycle process. So, specific genes that are involved with cystic fibrosis, there's only one gene, that is the CFTR gene, which encodes for the CFTR protein. And CFTR is a single gene loss of function mutation that causes cystic fibrosis. So losing that gene pretty much guarantees you that you would get cystic fibrosis. And the CFTR proteins encodes a 1,480 amino acid sequence protein. And this is publicly, something that I thought was pretty cool was this is publicly available in sequence databases found online. And then how you can test for this, that uses, and we'll discuss more about this later, but you could test, genetic testing can be completed that uses immunoreactive trypsinogen with reflex DNA testing common for CFTR variants. So, diagnostic methods. Every U.S. Newborn receives a IRT DNA based cystic fibrosis screening. And this is done by the gold standard, which is a quantitative sweat chloride test. Um, and genetic testing of the CFTR gene to identify pathogenic variants is used to confirm diagnosis. So, for the treatments and outlooks for management of CF, carrier screening is offered routinely to people planning to be pregnant, or to have a child. can reduce the incidence rate of CF. Um, for individuals that do have CF or cystic fibrosis, therapies include airway clearance, inhaled nebulizing medications, aggressive antibiotic therapy for infections, pancreatic enzymes, um, nutritional support, and in severe cases, a lung transplant. And for research, the outlook of research that is happening currently, it's ongoing. But one promising article that I found was the use of CRISPR, which is gene editing, pretty much, and it's aiming for a one-time and curative correction that is in active preclinical and early clinical development of this. So very promising results yet to come. And these are my citations, and I deeply appreciate it, and I hope that you learned something today. Thank you.